Hello, I'm Adriana Trotta, and today I'm going to present this work of my research group, which is about antimicrobial resistant patterns of clinical and commensal bacteria isolate from Loggerhead C. tartar. The overuse and misuse of antibiotics in human and veterinary medicine, as well as in agriculture and aquaculture, has contributed to increase the selective pressure on bacterial population, with the consequent appearance of new resistant strains and the dissemination of antimicrobial resistance in virus environments, such as the marine one. The Mediterranean marine water is the most waste polluted in Europe. In particular, the accumulation of resistant microorganisms involves the coastal seawater with shallow bottoms, such as the Adriatic Sea. The European Union Marine Strategy Framework Directive supports the research on wild sentinel species such as Loggerhead tartar for biomonitoring the state of marine health, assuming that their health reflects the health of the ecosystem where they reside. Loggerhead tartar have no history of antibiotic therapy and for their ecological and physiological characteristics tend to bioaccumulate contaminants such as toxin, pathogen, and heavy metals. In addition, high slate fidelity in the adult and juvenile life stage makes this species a perfect candidate for sentinel programs in the Adriatic Sea. Bacterial infection caused by gram-negative opportunistic pathogens are commonly reported in free-ranging and captive sea turtle. Many of these microorganisms are part of the sea turtle microbiota, but can be dangerous not only due to their opportunistic behavior, but also because they can harbor and share resistant genes, such as those conferring resistance to beta-lactams. In fact, in gram-negative bacteria, resistance against beta-lactams is mostly related to the production of hydrolactic enzymes, the beta-lactamases, and the extended spectrum beta-lactamases, ESBLs, which cleave the beta-lactams rings. Since these genes are located on plasmids, they can be easily transferred to other bacteria. Few studies have compared the bacteria sampled from free-living and hospitalized loggerhead or have documented the presence of antimicrobial resistant isolate from croacal and oral swabs of free-living turtles. Additionally, in the Mediterranean area, most of the studies have focused on antimicrobial resistant bacteria in loggerhead living in the western side of the Mediterranean Sea, creating a gap in their surveillance of loggerheads living in the Adriatic Sea. To fill this gap, we have recently published two studies to identify and compare the normal microbial community isolated from oral and cloacal sample of caretta caretta, healthy caretta caretta, with those obtained from internal and external wounds of inured animals. In addition, the presence of beta-lactamases and ESBL genes in isolate of inured subjects have been investigated. A total of 200 juvenile, subadult, and adult loggerhead sea turtles found stranded or accidentally captured by local fishermen during the winter season from the western Adriatic Sea of Apulia were included in the study. The animals were all housed in the sea turtle clinic of the Department of Veterinary Medicine during 4 years. All the turtles were brought by fishermen to the Centro Recupero Tartarughe Marine WWF di Molfetta and then to their sea turtle clinic for routine checks. In order to assess the health status of the subjects, each animal was routinely examined by veterinary personnel upon their arrival at the sea turtle clinic. In addition, a radiographical investigation was performed to check the pulmonary status. All the animals were hospitalized in the sea turtle clinic for a variable period of time, from 24 hours for healthy subjects until complete recovery for unhealthy or injured subjects. From each of 98 healthy turtles, 1 oral and 1 cloacal swabs were collected in order to investigate the commensal microorganisms. From the 102 injured turtles, swabs were collected from external or internal wounds, such as skin wounds, bulb, internal biopsies, Calabax wounds, or plastron wounds. Swabs were cultured and identified with biochemical micromethod tests and the 16S rRNA gene amplification and sequences were compared to those available in the GenBank database. The isolate were tested with the Kirby-Bauer method for the susceptibility to 8 antibiotics. isolates from oral and local sample were statistically compared with clinical swabs of inured animals for the proportion of isolates identified at genus level and the proportion of isolates of resistance against antibiotics. In addition, isolate from inured animals with reduced susceptibility to cephalosporins were screened for the presence of ESBLs by the double-disc synergy test and by PCR for the presence of genes encoding the ESBL, CTXM, SH2, and TAM genes. In addition, the Ambler class C cephalosporinases and PC genes were also investigated. In total, 280 commensal bacteria were collected from healthy turtles, while 130 bacteria were isolated from wounds of injured animals. When comparing the prevalence of genera of both groups, significant differences were found only for the Citrobacter genus, which were more prevalent in clinical samples. Several genera, such as Serratia, Moraxella, Culivera, or species such as E. coli, were isolated only in healthy animals, while Acinetobacter, Enterobacter, Klebsiella, Bacillus, and Morganella were isolated only in inured animals. When tested for susceptibility to antibiotics, clinical isolates showed highly significant differences in antimicrobial resistance rates compared to commensal isolate for most of the drugs tested. In total, 50 isolates displayed a multi-drug resistant phenotype. 13 isolates were positive for at least one resistant genes, and the variants of ESBL genes detected were BLA CTXM3, BLA TEM236, and BLA SH12. The variants of MPC belonged to the ACT, DHA, and CMY families. In addition, 4 isolates were found simultaneously harboring two genes, while 2 strains harboring 3 genes together. Bacterial infection by gram-negative seems to be one of the major causes of infectious disease in sea turtle, with high grades of morbidity and mortality. In our study, the same genera were isolated from healthy and inured animals, confirming that those organisms may act as opportunistic pathogens, depending on concurring factors such as parasitic, infectious, or traumatic injuries. More microbial species were found in the oral and cloaca swabs than in swabs collected from lesions. The antimicrobial resistance of bacteria from wild animals that have never been treated with antibiotics represent another interesting finding. In the present study, all the isolates collected from both groups were found to be resistant to at least one antimicrobial category, with very high resistance percentage, in particular against beta-lactams. Our study underlying as the ESBL genes identified in wild animals may be the same as those described in human isolates, therefore calling the attention on interspecies transmission mediated by successful ES-producing clones and plasmid among human, domestic, and wildlife species. The role of sea turtle as a reservoir of antimicrobial resistant strains should be deeply investigated, and particular attention should be given to the subpopulation of Caretta caretta. In fact, the Mediterranean subpopulation of Caretta caretta is poorly connected with the Atlantic one, and for their site fidelity, juvenile and adult subjects of Caretta are considered to be optimal ecological indicators of this zone. The detection of antimicrobial resistant rates in locales and the spreading of ESBLs producing microorganisms is of clinical and microbiological significance since it impacts the choice of a proper antibiotic therapy. So the antimicrobial susceptibility testing is recommended to guide the treatment of any bacterial infection. Lastly, systematic surveillance on these species should be implemented as well. Thank you for the attention.